Assalamualaikum and welcome back everyone. I am here for you to give a lecture on our course for chapter number 13. As you can see on the screen, our topic today is plant responses to the stimuli. I have divided my presentation into 3 parts. So that's my plan for this topic lecture. Let's get started. Okay, based on this slide again, I will begin my presentation with brief introduction for the topic, which is plant responses. So, plant responses. Like all organisms, plants detect and respond to the stimuli in their environment. So, what do we mean by stimuli here? Stimuli are referred to the changes in the external and internal environment of an organism, such as plant in our case. Unlike animals, plants cannot run. They cannot fly, they cannot swim toward the food or away from the dangers. They are usually rooted to the soil. Instead, a plant's primary means or response is to change how it is growing. Plants also don't have a nervous system to control their responses. Instead, their responses are generally controlled by the hormone, which is the chemical messenger molecule. For example, figure in this slide, shown plant shoot part or that we call polyptil exhibit phototropism response or growing toward the light source. Now we want to discuss about this slide, the learning outcome that derive from this topic. Specific learning outcome for the topic. Upon completion of this topic, students will be able to know plant response in axis movement. That's the first learning outcome for this chapter. The second one, plant response in active movement. Next, the third one, plant tropism in reaction to different stimuli. Then we also can gain a knowledge about the plant photomorphogenesis. The fifth learning outcome that can be originated from this topic, which is the plant photoperiodism and flowering. And last learning outcome for this topic, which is plant response to the environmental stresses. In the introduction part to this topic about understanding of the plant responses to the environmental stimuli. For example, plant roots provide structural anchorage to the ground on the soil and access to the water and nutrients, which is vital to the plant's survival. Therefore, must respond to the changes properties of the surrounding soil. Other example of the plant response toward environmental or external factor is the bending of the grass settling toward the light as shown in this figure, this one. It begins with the plant sensing the direction, quantity, and the color of the light as it stimuli. First response of the plant towards stimuli that we want to discuss for this topic is axis movement. Axis movement in the plant meaning is transfer movement of all part of the plant to the stimulus, that its direction is determined by the stimulus. Axis movement toward stimuli that we call positive axis. But if that axis movement away from the stimulus, that movement that we call as negative axis movement. This response movement consists of two kinds. The first one, which is photostasis, the second one that we call chemostasis. Based on this slide, photostasis, this is the first type of axis movement in plant. So, photostasis in terms of meaning, as a kind of stasis involves the movement of an organism or the whole their body that refer to the plant, which in response to a light. For example, among plant organelles that we call chloroplasts changes their position in response to the light. This mechanism that we call as chloroplast photorelocation movement, but their movement is not so obvious. Photostasis has been reported in many bacteria and also can be observed in blue-green algae. Next, organism whereby we can see the movement that we call photostasis inside the organism that we call diatom, include for desmin and other organisms which can produce the movement that we call photostasis, which is for many flagellates. Now we move to the next slide about chemostasis. So chemostasis is the second type of stasis movement in plant. So movement of organism in relationship to the chemical agent. So this is what we mean by chemostasis movement. So chemostasis involves the movement of plant towards or away from certain chemical response. So that kind of chemical response can be act as chemoattractant or can act as chemorepellent. For example, movement of spermatozoa or the mouse infant to ovule that is found in the archegonium, attractant to the sugar or protein molecule that is presented in the archegonium. So archegonium is referred as a female structure belong to the fern plant or to the mouse plant. Let's continue our lecture for this slide about nasty movement. So nasty movement is the second group of plant responses toward external or internal responses. Okay, now we want to discuss about the meaning for the nasty movement. So nasty movement in terms of its definition, the movement of a plant part or its organ only, not the whole body belongs to the plant. Okay, in response to an external stimulus, in which the direction or response is not determined by direction or stimulus. For example, for the nasty movement are in the Mimosa pudica plant, when we touch the leaf of the plant, they fall up. Here the stimulus is touch. The second example for nasty movement for the plant, which is in the dandelion flower. The opening up of the petal below the flower, okay, in the morning in bright light and closing in the evening when the light fades. Here the stimulus is light. Okay, next characteristic about nasty movement which is not regulated by hormone molecule. Next information related about nasty movement, temporary and reversible in terms of iteration. So it's meaning that in terms of it responds only for short-term period of time. This kind of response, as I said just now, involves of certain plant organ or plant parts, which are flower and leaf. This kind of movement again, okay, consists of two types. The first one that we call photonasty, and the second one that we call thermonasty. Okay, let's continue our lecture about photonasty. So photonasty is one of type nasty movement in plant. So in terms of meaning belong to the photonasty terms, photonasty is the movement of plant organ in response to the changes in light intensity. But direction of the stimulus in the form of light does not determine the direction of the movement of the plant. Figure in this slide, this one shows one species of plant that we call as Oxalis triangularis. This one. Now we want to discuss about the response of this plant when it will produce the response that we call photonasty. For the plant that we call Oxalis triangularis, is a great example of the plant that exhibits photonasty behavior. It leaves open up this one. For example, for the figure for this plant species at the upper part, okay, in this slide, okay, it will open up its leaf horizontally, okay, in response to the light. But if we close, okay, its leaf, for example, this one figure at the below part of this slide, if we close it will close its leaf like an umbrella during at night. Note that in this induced movement, depending on the light, there is no relation between direction of the light and direction of leaflet movement. From whatever direction you give the light, the leaflet belong to this plant that we call Oxalis triangularis will arise 90 degrees from the axis. Okay, let's continue our lecture on second type of nasty movement in plant. This kind of nasty movement that we call thermonasty. In terms of its meaning for the thermonasty movement, it is a such movement in the plant occur due to the change in temperature in the environment. Flower of the plant open with the rise in temperature, and that flower for the certain plant species, if we close its petal, with a drop in temperature. Figure in this slide, figure belong to this slide. This is the figure. Okay, shown the molasses movement belong to one flower species, that kind of flower that we call as crocus flower. This flower, okay, when in cool condition, it will close its petal. This is a little flower that we call crocus flower. It will open its petal during at warm condition. Crocuses open their petals as temperature rise and close them when it's getting colder. Sometimes repeating the action in between open and close, okay, in several times during the day. This movement protects the pollen from the snow and the rain, covering it until warm weather bring out pollinators. Now we want to discuss the third group of flower response that we call tropisms. So this is the next essential response group of the plant to the environmental stimuli that we call tropism. So in terms of meaning for the tropism response is the plant organ movement in response to the an external stimulus in the environment. If the plant organ moves toward a stimulus, then it exhibits positive tropism movement. If the plant moves away from the stimulus, this kind of movement that we call negative tropism movement. The growth, response for the plant when it produces tropism movement is governed one hormone molecule that present in plant tissue. Continue our lecture on tropism. There are 6 common tropism responses in the plant. The first one we call phototropism, second thigmotropism, third one we call gravitropism, the fourth for the tropism movement or response that we call hydrotropism. Next for the thermotropism, and last type of tropism mechanism that we call aerotropism. The first crude scientific experiment or study on plant phototropism was done by Charles Darwin and his son in 1880. Further explore about the research on phototropism done by Peter Boyson and Jensen in 1930. In the next slide, we will discuss in detail about Charles Darwin's experiment or study and the history behind the phototropism discovery. Figure in this slide show 2 experiments or study done by 2 group scientists on plant phototropism. Okay, the first experiment, this one, belongs to Darwin and his son in 1880, and this is another study or experiment done by another group of scientists, which is belong to Boyson and Jensen in 1930. Okay, in an experiment done by Charles and his son in 1880, they removed and covered part of the grass collective to determine which part sends the light. But in 1913, study by Peter Boyson and Jensen, they separated collective with different material to determine how the signal for the phototropism is transmitted. Result obtained in Charles Darwin and his son found that covering the tip with an opaque cover preventing the bending of the plant toward the source of the light. They also determined that the bending of the stem toward the light or the response to the stimulus occur lower below the tip. Then, Peter Boyson Jensen found that in their study, the phototropic signal from the tip of the plant is mobile. His experiment involved removing the tip of the stem and putting a gelatin block in between the tip and the rest of the plant. Since this block allows chemical to pass through, the plant produces a phototropic response toward the light. When he separated the tip and places a piece of mica or a piece of rock in place of gelatin block, nothing happened. The plant did not curve toward the light, as its normal would, because the chemicals that are moving in the stem cannot pass through the root.